Thank you very much. Thank you for the kind introduction. And it's a great honor to be here, especially because I like very much to talk about science to young people and probably future scientists. So let me tell you a few words about cellular friends and enemies in the context of brain metastasis formation. But let's start from the beginning. What is brain metastasis? So in the organism, there is a primary tumor, and some tumor cells spread through the bloodstream to the brain and form secondary tumors here, and these are called brain metastases. Very few tumors can do this, and this is a good news on the one hand, and I show you some other good news in the beginning. So these types of tumors are usually easily detectable in the beginning. Why am I saying this? So one type of tumor that forms brain metastasis is melanoma, which is a skin cancer. So you can see it. You can detect it from the beginning when it is still treatable and doesn't form metastasis. Similar with breast cancer, although we don't see it, we can basically touch it, so detect it very early, and then it is easy to cure. A little bit different is lung cancer because it is hidden in our body. However, by avoiding smoking, we can do a lot for not having lung cancer. So this is the good news. Now, let's move to the bad news. On the other hand, if we developed brain metastasis, the prognosis becomes extremely poor, so the survival time is less than 1 year. Right? So this is a really very bad news. And why is this? One reason is that therapeutic agents do not reach in relevant concentration these metastatic tumors because of the so-called blood-brain barrier, or BBB. What is this blood-brain barrier, or BBB? This is practically the vessels formed by the vessels of the brain, which are a little bit different in comparison to vessels of peripheral organs. Why am I saying this? So all vessels in our body are lined by the so-called endothelial cells. This is the same in the brain as well. However, here we have some specific other cell types that induce barrier properties in brain endothelial cells. These cell types are the so-called pericytes, which are perivascular cells. I'm a little bit lying because we have pericytes in the periphery as well, but here there are more of these cells. And the other cell type which is involved in this process is the so-called astrocyte, which is a brain-specific cell, is a star-like glial cell, which has very many functions. And one of its functions is to form these end feet, which are covering the capillaries, the vessels of the brain, and induce barrier properties in these endothelial cells. What are these barrier properties? On the one hand, endothelial cells in the brain are very tightly interconnected with tight junctions, which are kind of zipper-like structures. So these structures hold together endothelial cells in a very tight manner and do not let substances cross from the blood to the brain between the endothelial cells, because we have this zip-like structure between the endothelial cells. Not even ions can pass between the cells in the brain. Now, if a molecule cannot cross between the cells, it will try to cross through the endothelial cells, but here we have other barrier mechanisms of cerebral endothelial cells. One of these is the so-called efflux pumps, or efflux transporters, which pump back the substances that enter the endothelial cells, pump back into the bloodstream, so they cannot enter the brain. Why are these barrier mechanisms formed? Because the brain has to be protected from potentially harmful substances. Just think about ions. If they just get into the brain, they might interfere with the firing of the neurons. So this is a protective mechanism. However, this prevents the majority of therapeutic agents to get into the brain, not only really harmful substances, and this is why brain metastasis and other brain diseases are very hard to treat. Now comes the question, if this barrier is so restrictive, how can nutrients, for example, get into the brain, there are certain mechanisms, specific particular mechanisms for this. For example, glucose, which is needed for the neurons, does not freely cross the blood-brain barrier, no. But it has a specific transporter, a glucose transporter, which transports glucose from the bloodstream to the neurons, so it gets into the brain. Now, although this blood-brain barrier is really very strict and restrictive, there are certain molecules that can freely cross the blood-brain barrier, like this and this. I'm sure that everybody recognizes these molecules. That just for the sake of complexity or comprehensivity, I disclose. So this is caffeine. Let's say in the morning coffee that you drank, you probably, if you are a coffee drinker, you might have felt that it really helps a lot. So it has to cross the blood-brain barrier to reach the neurons. And the other one is ethanol, alcohol, which we heard about a little bit today. So it does cross the blood-brain barrier freely. It doesn't have any transporter. Okay, so as a coffee drinker, I say that this is the good. I need it in the morning. Although Zsuzsa was telling something different about the alcohol, I still say that this is the bad. If I may, I have to disclose that she wasn't drinking alcohol last night, I was. But anyway, let's say that this is the bad. And according to the Western movie, if we have a good, we have a bad, then we must have an ugly as well. And this is, in my case, the metastatic disease, so the brain metastasis. So although these substances can cross the blood-brain barrier, but we can say that the blood-brain barrier is really restrictive. Not even ions can pass through, as I said before. So how are then the tumor cells able to cross? Because this is the way for them to form metastasis in the brain. So they have developed two different methods to cross the blood-brain barrier. The first one is very obvious. As I told you, between the cells we have these tight junctions, the zipper, and some tumor cells can open this zipper, so they release factors which dissolve practically the proteins that make these tight junctions, and then they can move between the cells, so from the bloodstream into the brain to form the metastasis. However, there is another, a little bit more unexpected method of the cells. So these cells, which are not able to open the zip, so the zipper remains closed, they choose the transendothelial pathway, so they cross a single endothelial cell, and this is shown here. So what you see here is two endothelial cells here. This is the tight junctions, the zip, it's closed, and this is the tumor cell that is crossing this endothelial layer and is moving through one single endothelial cell, leaving the tight junctions intact. So these are the two mechanisms that the tumor cells are using to cross the blood-brain barrier. But very few tumor cell types can do this. So we can ask, is this blood-brain barrier, are these endothelial cells friends or enemies for the tumor cells? So on the one hand, they have to overcome these barrier properties. So here you see the green is the endothelial cell, is a brain capillary in a living animal. This red is a tumor cell in the phase of crossing the blood-brain barrier, so coming out from the vessel into the brain. And it has to overcome this barrier properties, so either open the tight junctions or squeeze somehow through an endothelial cells. So therefore, I can say that here the blood-brain barrier is a foe, is an enemy for the tumor cell that has to be overcome. On the other hand, when the tumor cells already get into the brain, like here, so the green is again the vessels and the red are the tumor cells, they are already behind the blood-brain barrier and protected from the therapy, right? Because the blood-brain barrier is restrictive and does not let drugs to get into the brain. Moreover, as you can see here, the tumor cells are growing around the vessels surrounding them. Why? Because they provide nutrients for them. So here, the vessels, the blood-brain barrier, is a friend for the tumor cells, and this we call a Janus-faced attitude. So Janus is an ancient god having two faces, a bad face and a good face, and if they can overcome, the bad face of the Janus, then they will take advantage of the positive effects of the blood-brain barrier. And the same was with other brain cells. So other cells in the brain have also Janus phase attitude towards the tumor cells. And I show just one example. It's quite frightening because it's very complicated, this image. But what you can see here, this is an astrocyte, and this is a tumor cell already that has already passed the blood-brain barrier, and this is inside the brain. And tumors and astrocytes first start to fight against these tumor cells, and therefore many of them die in the brain. Although they overcame the blood-brain barrier, they still can die because of other mechanisms, for example, because of some mechanisms related to astrocytes. However, some tumor cells can not only overcome these destructive mechanisms, but can even kind of reprogram the astrocytes to be protective for the tumor cells. And we found such mechanisms, for example, that I'm showing here on this cartoon. So we were examining brain metastatic triple-negative breast cancer cells, and these cells which after getting into the brain, they release some factors which induce astrocytes to release other factors, inflammatory factors. This is interleukin-1 beta, an inflammatory cytokine that induces a rapid proliferation of the tumor cells. So they kind of reprogram the astrocytes to become protective and release factors that induce proliferation of the tumor cells. And we found other mechanisms. So these are the astrocytes, you might remember, and there are these pericytes also around the vessels. And we found that they don't have to be reprogrammed. They already, from the beginning, release factors. This is a growth factor that induces proliferation of the metastatic cells. So this is another reason why the tumor cells grow along the vessels, because they have these pericytes there which secrete factors that they need for their proliferation. So these are really bad news. On the other hand, if we figure out these mechanisms, we might find novel targets. So these can be theoretically at least inhibited, and not only theoretically, in the practice, we found inhibitors that can stop these mechanisms and therefore inhibit these really rapid growth of the tumor cells. And we were really very lucky because both these inhibitors were kind of similar to caffeine or ethanol in terms of crossing the blood-brain barrier. So these, by their chemical structure, can freely cross the blood-brain barrier. So we were really very lucky in this case. And by using either of these, we could reduce the number and the size of the metastatic tumors in the brain of our experimental animals. Okay, so coming to a conclusion, I hope I could convince you that brain cells have a Janus-phased attitude towards the metastatic cells, being both foes and friends at the same time, and that we already know that brain metastases have a very poor prognosis at the moment, but we have to develop new therapeutic approaches. And hopefully we are on the good way to do this. And this is, I am optimistic not only about our results, but there are other results in the literature. And after we published our data about the astrocytes and the interleukin-1 beta and this inflammatory mechanism that I was talking about, a pharma company was highlighting our results on their website because they are developing similar inhibitors of this type that might be useful in the future for brain metastasis treatment. So in the end, I would like to thank my colleagues who were doing all this work. This is the Neurovascular Research Unit group in the Biological Research Center here in Szeged, led by István Kriszbai, and we are many people, including these. I don't see, Isti is also there, so including Szent Györgyi students, and thank you very much for your attention.
Thank you very much, Imola, for the beautiful and very inspiring presentation, and there is time for a few questions. Who has any questions? Please don't be discouraged.
While others are thinking, so you've shown very nicely that actually tumor cells transmigrate the endothelial monolayer via different routes, either passing at the junctions or tight junctions or through endothelial cells. Do we know about the chemokines that recruit them, whether they can be blocked, or what is your take on that?
So in terms of chemokines, we are we don't have really many information. But for instance, the IGF-2 that I was showing that pericytes are releasing kind of functions as a chemokine for the tumor cells. So they really sense it and move towards this. So it is not a chemokine, but a chemoattractant factor, I would say.
Any more questions? Please raise hands. Okay, there is one question in the back.
So I would like to say thank you for the presentation, and I would ask that wouldn't these chemotherapy toxins, basically, passing the blood-brain barrier cause more harm than good?
Oh, I see. So yes, the blood-brain barrier has been developed to protect the brain from all these potentially harmful substances. So there are some cases where incurable tumors are in the brain, and the blood-brain barrier, they try to open the blood-brain barrier so that the chemotherapy can get it. But this opening of the blood-brain barrier must be, limited in time, so because yes, the blood-brain barrier is there to protect the brain against the harmful substances. But if you have a tumor in the brain, you have to decide what you can gain and what you can lose by leaving these agents cross the BBB and do their job there. So in terms of these two specific inhibitors that we were using, they didn't cause any obvious harm for the animals by getting into the brain.
Okay, thank you.
Okay.
Any other questions? If no more questions, thank you very much, Imola, but don't go away, please, because we are handing you the certificate.